let us talk about the mechanisms of the antibiotic resistance. This is a topic of the practical importance because nowadays the day by day the antibiotic resistance is increasing by many bacteria. Uh, Due to some reasons, the antibiotic resistance is increasing because of inappropriate use of the antibiotics and the non-compliance of patient and the improper dosage of the antibiotics. These are the reasons. Because of these reasons, the antibiotic resistance is on the rise. So our topic of importance is the mechanism of antibiotic resistance. Why the resistance to the antibiotics is increasing. So our focus is the mechanisms. What is the antibiotic resistance? It is defined as a relative or complete lack of effect of antibiotic against a previously susceptible microbe. This means that there is a resistance towards the antibiotic. The previous microbe or the bacteria was susceptible, but it is gaining the resistance against the antibiotics. So what is the antibiotic resistance? It is the lack of the effect of the antibiotic against a previously susceptible microbe. Mechanisms of resistance. There are the four major mechanisms which are playing their role in the antibiotic resistance, and these four major mechanisms are inactivation by the enzymes and the alteration of the target sites. The target sites, they are modifying, they are mutated. That's why the resistance is developing. The third way is decreased permeability of the antibiotics, means the influx is decreasing. Entry of the antibiotics is decreasing. And the last one, the fourth one, is the efflux of the antibiotics, means the escape of the antibiotic from the interior of the bacteria. So they are rapidly escaping from the bacteria. So both of these, the influx and the efflux. Influx means entry and the efflux means the exit. So the entry is decreasing of the antibiotics and also the efflux is increasing. The influx is decreasing, efflux is increasing. Now coming to the first one, that is the inactivation by the enzymes. Let us describe these enzymes which inactivate the antibiotics. So which are the enzymes which inactivate the antibiotics? These are the enzymes. The beta lactamase. In the previous lecture, I elaborated what were the lactamases or the penicillinases. So the beta-lactamases, these are the enzymes which are present in some certain bacteria which inactivate or which degrade the beta-lactam ring of the penicillins, beta-lactam ring of the antibiotics. Beta-lactam ring is present in the antibiotics. So those bacteria which possess the beta-lactamases, they degrade the beta-lactam ring of antibiotics. So which are the antibiotics which are degraded or inactivated by these beta-lactamases? These are the drugs which are inactivated by the beta-lactamases because these antibiotics contain the beta-lactam ring in their structure. So one of the mode of inactivation of antibiotics is the beta-lactamases. Thereby, the antibiotics, they are degraded, and the resistance by the bacteria is developed. The second mode of increasing the antibiotic resistance is the modification of targets. The targets whereby the antibiotics act, these targets on the bacterial targets, they are mutated by these bacteria. They are changed, they are modified, they are mutated. Thereby the antibiotic resistance is developing. So, which are the targets which are modified or mutated? The following are the targets of the bacteria. These are the sites of the bacteria which are modified or mutated. The mutations occur in these sites, thereby the antibiotics do not work properly, and the resistance is developed towards them. So number one is the ribosomal unit, that is subunit 30S. This is mutated. Ribosomal subunit 30S is mutated, and the resistance develops towards the aminoglycosides. So when the ribosomal subunit 30S is mutated, the antibiotic resistance develops towards the aminoglycosides. And the protein binding proteins, the penicillin binding proteins, the penicillin binding proteins, these are the sites where the penicillin drugs are they act on these receptors. So these binding sites, penicillin binding sites, they are mutated. In this way, the penicillins become inactive, and the resistance towards them is developed by the bacteria, certain bacteria. And the third way is the DNA gyrase enzyme. This is the enzyme which is possessed by the bacteria, DNA gyrase, and those antibiotics which act, which inhibit the DNA gyrase, these are resisted, and towards the quinolone drugs, because the mode of the action of the quinolones is to inhibit the DNA gyrase. So in this way, if the mutation acquired in the gene which encodes for the DNA gyrase, the resistance develops towards the quinolones. The RNA polymerase is another enzyme, and the genes which code for this enzyme, in these genes, the, the mutation occurs, so the resistance develops towards the rifampicin drug. And the last one is the catalase peroxidase. These are both are the enzymes which are present in certain bacteria, especially the gram-positive bacteria. The catalase I described in the prior lecture, that how the catalase enzyme degrades the hydrogen peroxide. This is the enzyme which inactivates the hydrogen peroxide, which is produced during the reactions inside the bacteria. So those bacteria which possess the catalase, these inactivate these oxygen radicals, hydrogen peroxide, and the peroxidase enzyme, it is also inactivated. uh, and the superoxide radical, which is also inactivated by the peroxidase enzyme. So the hydrogen peroxide is inactivated by the catalase, while the superoxide radical is inactivated by the peroxidase enzyme. So when the mutations which encode for these enzymes, the the they are the mutated, those genes which encode for these enzymes, these are mutated, and thereby the resistance is developed towards the isoniazid. Isoniazid is a drug which is given in the tuberculosis. So these are the targets where the mutations occur, and the different types of the antibiotics, they become inactive, or the bacteria become resistant to these antibiotics. So these targets are modified. Third way is the reduced permeability of the antibiotics. So when there is a reduced permeability, there is a decreased influx of the antibiotics. So there is a decreased entry of the antibiotics when the permeability, when the cell wall permeability or the cell membrane permeability of the bacteria is reduced, resulting into the decreased entry of the antibiotics. So when there is a decreased entry of the antibiotics into the bacterial cell, uh, and so in this way the antibiotics are no more effective, and these antibiotics are no more effective against these bacteria. So which how there acquires the reduced permeability, there is the porin proteins. There are the pores in the cell wall, the in the cell wall of the bacteria. These pores proteins, they are mutated. Porins are the pores which are present in the cell wall of the bacteria, by which the antibiotics, they are entered into the bacterial uh, bacterial inside of the bacteria, And when these foreign proteins they are mutated, the antibiotics, the bacteria gain resistance towards these antibiotics. So these are the antibiotics which enter by these pores, these porins. So when these are mutated, these porin proteins, they are mutated. These antibiotics, they are not entered efficiently inside of the bacteria. Fourth way of antibiotic resistance is the increased efflux. I think this is a decreased, increased efflux. So yes, increased efflux. So the antibiotics which enter inside the bacteria, they are uh, sufficiently escaped out from the bacterial cytoplasm. So they are efficiently escaped out. So there is a rapid escaping of the uh, antibiotics from the interior of the bacteria. And which are the antibiotics which are efficiently escaped out? These are the tetracyclines, sulfonamides, and quinolones. So there is a rapid escaping out of the antibiotics from the interior of the bacteria. And when the antibiotics they are escaped out, so how they can act on these bacteria? So this image shows these four before mentioned mechanisms of antibiotic resistance. The first one is the inactivation by the enzymes, and the second one is the here shows the efflux, the efflux of the antibiotics. They are escaping out from the interior of the bacteria. So there is a rapid efflux. And here is the alteration of the targets, and these targets are the DNA gyrase. Here is the decreased uptake, influx. This one is the decreased uptake, means the influx. Here is the decreased uptake, and here is the increased efflux. So these are the four mechanisms which the bacteria they gain by the mutations and by the different mechanisms. So and now coming to the molecular genetics of the antibiotic resistance, the how the genetically the bacteria gain the antibiotic resistance. The genetic variability is essential for the microbial evolution. Every bios, every living organism gets evolution with the passage of time. It adapts in the environment, and so this is called the genetic variability. The evolution. This is a process of evolution which occurs in every living organism. So, regarding the microbes, regarding the bacteria, the genetic variability is essential for the microbial evolution. It may occur in a variety of ways. So these are the methods, these are the ways by which the genetic variability occurs. How genetic variability occurs. These are the different uh, ways in the genes are altered. The genes, the nucleotide is a part of the DNA, and the alterations occurs in these genes, and these are one is the point mutation and the invariant. These are the mutations. Mutations do, do, do not occur only the human beings, and these also occur in the microbes. So I mentioned that the evolution and the mutations occur in every living organism. So the bacterial mutation, the bacterial genetic variability also occurs. So these are the ways by which the bacterial genetics varies and gains the antibiotic resistance. The basis of drug resistance. What is the basis? How the drug resistance, uh, on which basis it is to be mentioned? The drug resistance or the antibiotic resistance, it may broadly be categorized in two ways. One is the genetic drug resistance, and second is the non-genetic drug resistance. So the genetic drug resistance occurs by these three ways. The the it may be the antibiotic resistance may be the chromosomal mediated, may be plasmid mediated, and the transposons mediated. These are the some inside structure of bacteria. The chromosomal mediated, the 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 DNA or the chromosome of the bacteria. So it may be the chromosomal mediated, and what is the plasmid? Plasmid is also the structure inside of the bacteria by which these are the extra chromosomal structures or the segment of the DNA, and by these plasmids the drug resistance also developed. And what are the transposons? These are also the segments of the DNA. And remember, these transposons are called the jumping genes. Transposons are the jumping genes because they move from one structure to another structure inside the bacteria. So they are called the jumping genes. So the genetic antibiotic resistance also may be the transposons mediated. So these are the genetic ways by which the drug resistance is developed, and it may be the chromosomal mediated, plasmid mediated, and transposons mediated. Now coming to the non-genetic basis. Let us describe the chromosomal. First one was the chromosomal mediated, briefly describing the chromosomal mediated. The the chromosomal mediated drug resistance occurs as a result of the spontaneous mutation. So the mutations occur at the level of the genes, at the level of the chromosome, and thereby the drug resistance is resulted. So this is a cause by a mutation in gene that codes for their target cell, for the target of the drugs or the transport system in the membrane of the cell wall that controls the entry of the drugs into the cell. So mentioning the chromosomal mediated resistance, the this describes the how the antibiotic resistance is developed by the chromosomes. So this one is a genetic basis of the drug resistance. Second was the plasmid mediated, and the plasmids, these are also called the R factors, means the resistant factors. They produce the resistance towards the antibiotics. So it is mediated by the resistance plasmid, the plasmids which are labeled as a R factor, and these are known as the R factor. So this is one of the genetic basis of the drug resistance. And third way of the genetic basis is the transposon mediated drug resistance. So let us come to the non-genetic mechanisms. Non-genetic basis. Non-genetic basis of resistance plays a less important role in development of the drug resistance. The most of the drug resistance towards the antibiotics is developed by genetically by the mutations in the different uh, genes. So the non-genetic basis is less important. Certain bacteria produce the pus. In certain cases, the the certain bacteria under the ordinary circumstances are usually killed by the penicillins. But some bacteria, they are not killed, and they only lose their cell wall, and they are called to be protoplast. When some bacteria, when these certain bacteria, they are treated by the penicillins, they lose their cell wall and they become the protoplast. And protoplast is a atypical form of the bacteria. So in this way, it develops the drug resistance. The bacteria changes its shape and cell wall is lost. When the cell wall is lost, the certain sites, target sites, they are changed. The foreign proteins, and the these are mutated. In this way, the protoplast become non-susceptible to the action of the some cell wall acting drugs such as the penicillin. In certain conditions, the bacteria which are pus producing, or which are called the purulent organisms, purulent bacteria, they produce the abscess. Abscess, this is the form, accumulated form of the pus, that is that is surrounded by the wall, is called the abscess. So the certain conditions, in certain conditions, as in abscess, the bacteria can be walled off, which prevent the drug to penetrate effectively into bacteria. So in this way, the bacteria protect themselves and they resist to the antibiotics. So the antibiotics become ineffective when the abscess is formed, until and unless the surgical drainage of the pus is proceeded. So this is one of the mechanism by which the bacteria protect themselves and become resistant to the certain antibiotics. These in some procedures, if the foreign bodies, uh, they are within the living organism, such as the surgical implants, catheters, when they are kept inside the wound, and this way they are not degraded by the they are not affected by the antibiotics. So these foreign bodies and sutures, catheters, splinters, the antibiotics become ineffective in such type of the procedures. Thank you very much for listening.